His PhD from University of Pune. At CSIR Institute of Genomics and Interactive Biology, Delhi, India. Postdoctoral Fellow from University of Gothenburg, Sweden. Dr. Srinivas is Career Development Fellow at MRC Laboratory of Molecular Biology, Cambridge, UK. EMBO Long-Term Fellow at MRC Laboratory of Molecular Biology, Cambridge, UK. Investigator Scientist at MRC Laboratory of Molecular Biology, Cambridge, UK. And currently, Dr. Srinivas is Assistant Professor at Indian Institute of Science Education and Research, Tirupati, India. Dr. Srinivas got many awards and honors, like a few I'm mentioning here because the list is very big. And that UK Medical Research Council end of the year special award scheme for outstanding scientific contributions in 2017. The European Network NGP NET COST Action Short-Term Scientific Mission Fellowship to undertake a sabbatical at Institute for Molecular and Cell Biology, Portugal in 2018. Research Fellowship, Wolfson College, University of Cambridge. Associate Faculty Member, F1000 Prime in the field of Genomics and Genetics. MRC Special Award Scheme Light Award for contributions to public outreach during Cambridge Science Festival in 2015. FUBS Young Scientist Forum Fellowship in 2013, Indian National Science Academy Young Scientist Medal in 2012, elected member of Educational Policy Committee, Wolfson College, University of Cambridge, and also Dr. Srinivas got Junior Research Fellowship from Wolfson College, University of Cambridge. And also from Indian point of view, he is awarded this Junior Research pursuing PhD studies. Dr. Srinivas is editorial board member for Amino Acids, a Journal of Biosciences, a grant application reviewer for Austrian Science Fund, peer reviewer of nucleic acids research, molecular biology and evolution, bioinformatics, briefings in bioinformatics, BBA gene regulatory mechanisms, scientific reports, molecular omics, the FUBS journal, molecular biosystems, genome biology and evolution, ACS Omega, clinical genetics, protein science, PLOS1, evolutionary bioinformatics, gene, current genomics, Journal of Structural Biology, Journal of Theoretical Biology, Molecular Biology, Report Archives of Medical Research, and Journal of Bioscience. He has got a very good number of publications which crossed more than 2,600 citations with the H index of 26 and item index of 40. The main focus of his research is to elucidate the physiological and pathological consequences of variations in the structured region and disordered regions, that is low complexity sequences and repeats of proteins. For this, he employs approaches and the principles from genomics, systems biology, structural bioinformatics, and machine learning. Now, I request Dr. Srinivas Chavali to start the highly informative session of the workshop, that is protein structure prediction and network analysis by employing some softwares like PyReju, Swiss model, and Cytoscape. Over to you, Dr. Srinivas.
So we'll two sessions today. One on the prediction that will be done by my postdoc, Dr. Rashekar Verma Kadmuri, and subsequently I'll discuss on the network analysis. Raj, can you please start your presentation?
Sure.
So, yeah.
Thank you. Hello, and a very good afternoon. So today, first I'll share my screen. Are you guys able to see the presentation?
Yes.
Yeah.
So we get into full screen mode.
Yes.
I guess you have to flip it around. As we see the presenter view.
Yes.
That's good.
So
Can I move to the next slide?
Yep.
Yes, please.
So hello everyone. A very good afternoon. Today I'll be sharing with you guys how to infer the protein structure from a given protein sequence of our interest. So for this, we'll be looking at a technique called protein modeling. So experimentally predicting a protein structure is very labor and time expensive work. So because of this, computationally modeling a protein and predicting the structure will give us some heads up into the structural and functional aspects of that protein of our interest. So regarding this, today we'll be looking at a couple of things with respect to protein structure modeling. So what we'll be discussing, first we'll look at the protein structures and their basic principles, which will help us to understand the protein modeling in a very detailed manner. And then we will see the different levels of protein structures, secondary structures of the proteins, motifs, supersecondary structures, folds, and then I'll also introduce you guys to the protein data bank.
Excuse me, Dr. Rashekar.
Yes.
Can I take few seconds to introduce you to the participants?
Okay.
Just a few minutes. Just introduce Dr. Srinivas. Okay, thank you, Rashekar. Dr. Rashekar Verma, right now you are seeing, he's a postdoctoral fellow and he has he's also having expertise in bioinformatics and data mining and biological databases, data management, web page developments, and he completed his PhD in computational structural biology from BITS Pilani, Hyderabad campus, MSc Biotechnology from Bangalore University, PG Diploma in Bioinformatics from Anna University, BSc Biotechnology from Sri Venkateswara University, Tirupati. Okay, he's really have very good academic career. He got many fellowships and awards, served National Postdoctoral Fellowship. Right now he's on that fellowship, he's associated with ICER, with Dr. Srinivas Chavali. And also he got DBT Research Associate Fellowship, adjunct assistant professor in the Department of Biohealth Informatics at the Indiana University. He got Best Poster Award from ICER, BITS Pilani Hyderabad Campus Institute Fellowship also he achieved, and University UGC Fellowship also he availed. And he has so many publications in very reputed journals. And really, if you see his academic thing, he has very good expertise in biotechnology and bioinformatics. I hope all the participants can interact with Rashekar, and you can be in touch with him and clarify your doubts. I hope you all will enjoy Dr. Rashekhar's presentation. Over to you, Dr. Rashekar.
Thank you, Dr. Marathi. And thank you all. So we'll resume our presentation talk now. Yeah. Are the slides visible, right? So regarding this protein structure prediction,
and you have to flip. It's in the presenter mode.
Oh.
Yeah, it's good now. Please carry on.
So to start with, we'll be discussing about the protein structures, the basic principles, and the levels of protein structures, secondary structures, and the different types of secondary structures, which you'll frequently observe in the protein structures, and the different folds that the proteins get organized into. And I'll introduce you to the protein data bank, where all the experimentally identified proteins are deposited, and how to query for a protein structure, and the summary of the deposited protein structure. So what all information you can find, and the experimental studies that have been done regarding this, and the structural aspects of it, and the secondary structure content, sequence information, everything you can find at the protein data bank. And we'll look at the general protein modeling methodology, and then I'll introduce you to the PIRE2 homology modeling methodology, and also the ab initio modeling methodology. So then I will take you to the PIRE2 modeling server. It's an automated modeling server. So we'll be doing a live session here. I'll show you guys how to, given a sequence, how can we get a protein structure and how to analyze the results after that. And we'll also use this model. It's another automated modeling server. And then once we get the predicted models, we will also see how to do a quality assessment of the structures using one of the reputed server called NCBI Saves website. And once we get the quality assessment, we'll also see how to do a refinement of your structure. I mean, there are different servers available. So these servers mostly work based on the energy minimization part of the models. And I'll do a brief intro about this as well to you guys. So going forward, as most of us know, like the proteins are the peptide chains to start with. So amino acids form peptide bond. Okay, so they form a dipeptide bond from the N-terminal and C-terminal regions. So two amino acids come together, and the N-terminal C-terminal regions, the amide and carboxyl group, they come into form a dipeptide bond by releasing the water molecule. Now, once we have the string of amino acids coming together, it's called the polypeptide chain. And this polypeptide chain backbone conformation properties are primarily restricted or maintained by the dihydrolamines. Okay, so what are the dihydrolamines? So if you see here, this is a typical polypeptide chain of two amino acids. So if you see here, between the C alpha atom and C atom, you have a bond angle. Okay, so you have psi and phi bond angles coming up over here. So the one is between C alpha and C, and the other one is N to C alpha. And so these bond angles can permit a flexible rotation between plus 180 to minus 180. Okay, so you have a rotational conformation that can be accepted within this 180 to minus 180. So both phi and psi can rotate to within a range of plus 180 to minus 180. Now, so this will allow the backbone of protein, to acquire certain conformations with a little bit of restrictions. Okay, if you see here, so you see here, these are the two amino acids, and depending on certain bond rotation angles, the flexibility of the backbone changes a bit. Okay, with this, I would like to introduce you to something called Ramchandran plot. Okay, so Ramchandran plot, which is given by Dr. G. N. Ramchandran, will actually allow us to plot these dihedral angles, which lies within the range of minus 180 to plus 180. So for every amino acid, we have the phi and psi degrees, right? The dihedral angle degrees. So the rotation of these dihedral angles can be plotted onto this Ramchandran plot. So for the most of the known conformations, it was observed that they actually occupy certain regions in this Ramchandran plot. So there are mainly three regions. One is the most favored or allowed region, and the partially allowed region, and the disallowed region. Okay, so Ramchandran plot also plays a crucial, why I'm discussing about this is the Ramchandran plot also plays a crucial role in evaluating the model that you're going to predict. Okay, we'll see how we will do that. Okay, so to recap this, we have -180 to +180, which is the rotational restriction of the dihedral angle. So you can the atoms can only rotate within this stretch of degrees. Okay, so if you plot a psi and phi, you'll have a certain region that is being occupied by the residue. And as I mentioned, different protein conformations or the structures are known to occupy certain regions in the transition plot. So it also helps to validate the structures. Okay, so these are the experimental studies that have been done for a huge amount of which has consumed actually huge amount of time to come up with this kind of analysis. So all the experimental studies that has been done on the structures, open structures, are actually help to get to this information. So we take all the structures, we just plot, get the dihedral angles of them, and then plot them onto the transition plot, and then we see certain kind of conservation. Okay, that can be acquired by these structures. Okay, so going forward, so I would like to introduce to the different levels of the protein structures. As we know, we start with the polypeptide sequence, that is the strings of amino acids, which will fold to form a secondary structure content. We can be either helix or strands and the loops. Okay, which will again lead to the tertiary structure, followed by the quaternary structure. Now, as I mentioned, backbone conformation flexibility is managed by the dihedral angles. But what is the driving force to reach from the primary structure to the quaternary structure? So the side chain chemistry actually plays a crucial role in bringing up the protein structure. Okay, so to show you this, I have a small video over here. So if you see, this is a linear conformation, the polypeptide sequence, the side chain atoms shown here. And on the right, if you see, this is something called energy well. Okay, so one of linear conformation, protein linear conformation, is the starting conformation. So it is right here now. So when it starts to fold, what it will do, it will try to explore the energy states. So its main goal is to reach the lowest energy minimization state that is possible. So it has to sit in a very comfortable lowest energy state. So in this process, what it will do, it will try to explore all the possible, this well, it will explore this well, and goes through the all possible, conformations, which can favor for it to sit in the best energy well. So I'll just play the animation. You can go through it, and then I will talk about this again. So as you can see, we started from a linear conformation, and by the time it acquires the best possible conformation, it is in its lowest energy favorable state. So in this process, it is actually exploring. I'll make it go slow. If you see this black dot here, so this is the conformation, protein conformation, and this entire thing is the energy space. So it is exploring all possible states and all the possible, hollow wells and the possible steep wells. Okay, and then it is settling down in the best energy minimized state. Okay, so to brief you, the protein has to be in its best energy minimized state. So it should be in a very favorable position where it can have the best energy, lowest energy state. Okay. Now, so we'll talk more about this in the energy minimization part of the protein modeling. Okay. Now, so I'll briefly introduce you to the protein secondary structures as well. So to start with, we have an alpha helix, which once we have, we'll get an alpha helix that get folded, and we have strands. So there are two types of strands. One is like parallels and the anti-parallels. So based on their hydrogen bonding pattern, they are different into parallel and anti-parallel. And if you see within the structure, this is how they appear. So we have the strands that are parallel in nature, and we have strands which are anti-parallel in nature. Okay, so the N and C terminal directions will alter in the anti-parallel, and here we have them in the parallel. We won't go into the chemistry of these structures right now. So if anybody is interested, we can at the end of the session we can look into the chemistries as well. Now, so going forward, like from the secondary structures, the next information that a protein acquires, something called motifs or super secondary structures. Okay, so there are a number of motifs that one can observe, but I'll show you here the most frequently observed motifs. So one of them is EF hand, also called as alpha loop, alpha motif. So these kind of motifs are frequently observed in protein structures. So if you see here, this is a helix, and you have a loop and a small strand coming up over here with a helix, and you have a calcium ion binding over here. Okay, so these kind of motifs are mostly helpful in the binding patterns. And the next one is the Greek key motif. So in the Greek key motif, so you have four beta strands, in the anti-parallel, and then interjoined together from the N-terminal to C regions. So the third one is beta hairpin loop. So you have two beta strands forming a hairpin kind of conformation. Okay, so with a very short loop coming up over here. So again, beta hairpin loop is also involved in certain kind of interactions. And you have a beta alpha beta motif. Okay, this is also again a kind of motif where a strand is followed by helix and then again a strand. So if you see here, just to give an example, I've shown how a motif can be the part of a three-dimensional conformation. Okay, so if you see here, this is a timbaral protein, which is made up of 8 helices and 8 strands, which are alternating in nature. So you see, you have a beta barrel inside and followed by a helix shield. Okay, which is an amphipathic in nature. Okay, so this gives you a brief glimpse of how different motifs exist in the protein conformation or a structure, and every motif known to play certain roles, like it can be a binding, it can be some kind of small function, or ion binding, etc. Protein-protein interactions, it can be. Okay. Now, so from folds motifs, we'll look at the folds. Okay, which is a journey towards the tertiary conformation. So to start with, we have alpha folds first. Okay, you mostly see them in helix bundles or globin folds, homeodomain folds, and alpha solenoid structures. And you have beta barrel folds. Okay, you see them in again immunoglobulin domains, beta barrel or beta popular proteins, etc. And you have alpha by beta fold, so where you have helix followed by strand, and then again it will change to helix. Okay, so helix strand alternate. You see this kind of folds in timbaral, leucine-rich repeat proteins, cleodoxin folds, and Rassmann folds. So I have a cleodoxin fold protein here as an example. And we have one more fold called alpha plus beta folds. Okay, so they are mostly observed in DNA clamps, cleodoxin fold, ribonuclease, and SH2-like fold. So the main difference between alpha by beta fold and alpha plus beta fold is alpha by beta are alternating in nature, but whereas alpha plus beta fold, they are not. If you see here, so we have strand content followed by the helical content. Okay, so these are the 4 major classes of folds: alpha, beta, alpha by beta, and alpha plus beta. And when we go to the protein data bank, which is a primary database where all the protein structures are deposited, so the entire proteins are classified, the deposited proteins are classified into these 4 major classes. So we have a class of alpha folds, class of beta folds, alpha by beta folds, and alpha plus beta folds. Okay. Now, so I've been working on protein structures from decades, and there's a lot of inflow from the experimental biologists to predict a protein structure using different experimental techniques like XRD, NMR, or cryo microscopy, electron microscopy, or something. So all this information, it has to be shared with public, right? So, but where is it getting deposited? They have to have a certain central repository to share with everyone. So regarding this, people have come up with a primary structural database where you can actually deposit the structures, and these structures will be publicly available to everyone to work with. Okay, so here I'll present you guys one major database that is called Protein
Sorry,
Data Bank.
Raj, we are not able to see your slides.
Oh. Is it visible?
No, I guess. Can you move your slides back and
I
forth?
am.
Is it that only I have an issue? I just see a thin strip on your slide. It's nothing.
We also have same issue. No slides are visible.
So suddenly this has happened. Until now, we were able to see your slides.
Yeah.
Okay.
Yeah.
Now it's okay.
But this is in presenter mode, so you can get back to the main mode. Yeah. Please go.
Yeah. Are you able to see the?
Yes. Please go.
Yeah. Should I go back to the previous slide or?
This is fine.
Yeah.
You can get going.
So, okay. So we are looking at the. I will take you to the protein data bank, and we can actually. I'll show you to how to query for protein structures, and also some statistics over there. And if you see here, this is how the explosion in the data has happened with respect to protein structures. So if you see here from 1976 to 2021, actually there is a very big explosion happening over here with respect to number of protein structures that have been identified annually. Okay. So before I take you there, I'll just show you two slides where I can briefly show you the how to query. So if you see here, you have a search bar here. Here you can either give a text with respect to protein name or anything, or every protein that is getting deposited to protein data bank, it will be assigned with an ID called PDB ID. Okay. So if you know the PDB ID, you can directly enter the PDB ID to query, or you can enter the respective text. So it can be a kinase, or it can be specifically if you want to go, you can say arginine succinylsynthetase, or you can say protein kinase families, or you can just type the text there. Okay. And once you do that, you'll be presented with multiple results. Okay, so I just picked one over here to show you. So if you see here, this is how a specific result page looks like. You'll have this structure visualization happening over here. So this is structure snapshot, but if you want to look at it in a 3D mode, you can do that as well. And you have the PDB ID. So you have the PDB ID, and you have the name of the structure and the classification to what it belongs to, and the organism from what it has been predicted, and you have the deposited and the authors and information with respect to the experimental studies. So let's say if it is an X-ray diffraction method that has been used to get the structure, we have the resolution information, like to get the sensitivity of the the predicted structure, you can go through the resolution as well. And also you have certain stats with respect to the quality of the structure that has been deposited. Okay, so I'll stop at this point and I'll take you to the PDB, and I'll show you briefly like what we were talking about. So you guys are able to see the desktop, right?
Yes.
So this is the web page, PDB homepage that you can go to. So if you see here, we have on the first tab, we have certain information with respect to deposition, or you can search based on certain information. For example, like you can even use some tools to draw the molecules and search. You can do a sequence search, and you have some basic search, and you also have the PDB statistics, which will give you the information with respect to the deposited structures and everything. Okay, so if you see here, the PDB data distribution by resolution is there. If it is an XRD, okay, you can search by ligands, you can search by drugs or drug targets. Okay, and we can even analyze the data, pair of structure element you can do, protein symmetries you can check, or if you want, you can download the entire data sets as well. But we'll not get into the advanced search modes right now. So quickly, I'll show you an example over here how to search. So I just type. So I'm just giving a text over here, or I can also give a PDB ID. So to see here, so this is something called advanced search query builder. If you won't be getting into that right now. So this is how your result summary looks like. So you have multiple structures with respect to this name. Okay, so we have a number of structures, and this is how the result looks. So this is each entry is given as a tab. So you have the list of entries coming up over here. On the left, we have something called refinements. So what you can do, you can specifically filter these protein structures as per your need. For example, if you want the structures with respect to only Homo sapiens, you can just select the Homo sapiens and then search based on the Homo sapiens. Okay, and it got filtered right now. Okay, similarly, you can do filtering based on certain criteria, like if you want to put in the DNA or RNA, you can do that. I mean, if it is in complex with the DNA or RNA, or if it is in with respect to specific experimental technique, if you want to search, that also you can do. And if it is with respect to refinement resolution, so if you want to get a very high good resolution structures, that also you can do by opting for the best resolution that is available out there. You see, the results are getting trimmed down. Okay, so if I go ahead and click on one of the structures, so that will take me to the specific page of that respective structure. And if you see here, there is huge information that is available here. So let's talk about one by one right now. So this we have discussed. These are the quality statistics and the information with respect to the structure deposited. And if you scroll down, we have the literature that belongs to this respective protein structure. Okay, and we have information with respect to the number of chains that are there, the sequence length, okay, and the mutation information, disorder regions, everything. So all this information is for the known protein structures. Okay, so whatever protein structure that has been identified, or predicted through the experimental techniques, it's deposited here with the complete information. Okay, you can even go and look at the structure in a three-dimensional space. Okay, so along with the ligands, the information is deposited everywhere. And it has been deposited. If you want to play around, you can play around and look for the ligands and interacting regions, etc. You have the annotations available. Okay, so you have the complete information coming up over here. So the class to which it belongs, as I mentioned, alpha plus beta class. Okay, and you have the domain annotations to what kind of domain it belongs to, and the protein family information, the gene product annotations, okay, bindings, okay, any drug molecules that are included, biological processes that it is involved in, and cellular components that it has been related to, etc. Okay, so all this is for the known structures. But let's say if we have a sequence which you might have identified in your studies, which is coming from some experimental studies. So you might have observed that pathogen is interacting with host, and a specific protein is involved in it. And if you go ahead and do a sequencing of that protein, you'll end up with this sequence. But if you don't have a structure for that, going through the complete experimental protocol to get the structure for that sequence, it's a very time-consuming and very labor-intensive work. So to give a heads up, what you can do a computational protein prediction for that sequence, and get the some insights like how the structure is and to what kind of class or domain what kind of class it belongs to, and what kind of a domain it has, and what kind of functions it can get involved into, and the interaction that it can get involved into. All these kind of things can be explored if you can model protein structure using the computational modeling technique. Okay, so next we will see the protein modeling technique for the unknown sequence. So for this study, before getting into this, I will share with you guys the protein modeling methodology that people used to use traditionally, and I'll also share with you guys the automated tools that are out there. Okay, So I'll get back to the. So you guys are able to see these slides, right?
Yes.
Yeah. So the basic protein modeling methodology, we used to have a very traditional model methodology that has been followed by everyone. So each step has to be manually, which needs a manual intervening part. So you, for every step, you do it manually. So and at the end, we'll compile everything, and we used to get the protein structures. But the technology has advanced so much, and now we have the automated servers which can do everything for us at the back end. But I'll also walk you through the basic modeling methodology so that you have an understanding of what is happening behind the screen. Okay. So to start with, we'll have a query sequence of our interest, and we have to reach to this destination or the final model. Okay. So in this process, let's say you'll start with the protein sequence. It can be either in a FASTA format or it can be in a plain text format. So if you're ready with your protein sequence, what you'll do as the first step is to identify the template. So what are templates? So as I mentioned, most of the protein structures will have similar backbone conformations, right? Okay, depending on the protein sequence similarity. So for protein sequence is known to have more than 30% sequence similarity, or 35%, 30 to 35%. So it is tend to acquire certain structure that target sequence. So let's say if you have the sequence A and sequence B, and if sequence A he's having structures, sequence similarity of 50% with sequence B, both of them are tend to acquire certain structural similarity. Okay, so based on this concept, so what we do is to look for the known structures. Okay, known structures for which the sequence similarity with our query sequence is at least about 30 to 35%. Okay, so you need to look for the sequences which have structures, okay, that are at least within the range of 30 to 35% sequence similarity and a coverage of more than 70%. Okay, two sequences should cover at least in the range of 70%. Both of them should overlap. Should align together. Okay. So once you look for the known structural sequences for which the structures existing, you'll identify certain templates. But how to do this? Okay, so we have different methods to do this. Like traditional one is to do a sequence alignment. So you'll take your query sequence and you'll align it against the known sequences for which the structures are known. Okay, against the sequences for which the structures are known. So once you do this based on the sequence alignment, if it is meeting these two criteria, either 30 to 35% minimum range or 70% coverage, you will pick the respective template and use it as the target template for your protein modeling. Okay, so there are different alignment techniques available, as I mentioned. One is PSI-BLAST, okay, the position specific iterative blast, and the other one is hidden Markov model based searches. So both of them looks for the conserved regions within the sequence, and then they will retrieve the respective templates based on the sequence similarity. So once we do the sequence similarity search, then you'll end up with either one single template or with multiple templates. Okay, it's not necessary that you'll end up with only one single template. You'll end up with multiple sequences for which the structures are known. So I'll also discuss about this, whether you should go for one single template or multiple templates, how that is going to change your protein modeling. Okay, so now once you get the templates ready, you'll go for the further step and go ahead and do the modeling. But what if you don't have the templates identified? We'll talk about that in a second, don't worry. So we have the templates ready with us. We identified some templates. Okay, so if you have the templates identified, we'll go for the homology modeling. Okay, in the homology modeling, the first step is to do a backbone modeling. So as I mentioned, the backbone atoms, mainly the C alpha atoms, is the backbone of the protein structure, right? So what we'll do, we'll go ahead and do a backbone model query sequence for which the structure has to be predicted. So once we have the backbone confirmation ready, then we'll do something called loop modeling. Okay, so loops are the very difficult region that can be modified because these are highly dynamic in nature, and the sequence similarity that you find for these loops are also very low because they tend to change a lot with respect to evolution. So the loop modelings are like it's difficult to model in loop regions due to their dynamic nature and the less conservation in the protein sequence. So once you have the loop modeling done, you will do something called sidechain modeling. So the sidechain modeling is done based on certain sidechain rotamer libraries. And finally, you'll have minimization done for the entire structure that has been modeled to make sure that there are no steric clashes that are available, and the protein is perfectly sitting in its energy purity state. Okay, so with respect to this, we'll be discussing about PIRE2 and Swiss model, and there are other two softwares which can be used to fine-tune your models, like Modeler or Rosetta. Okay, so these are like they need human intervention at every step, and you can actually fine-tune to the atomic level in these two tools. But the PIRE2 and Swiss model are like the fully automated tools. You can just submit your sequence, and this entire steps, this entire process is actually fine-tuned and packed into one single package, which is available in the form of PIRE2 and Swiss model servers. Okay, so now if you have templates, that's well and good. You go for homology modeling, and everything is fine. But let's say if you don't have templates, no protein structures have been identified till now which has a sequence similarity with your query sequence. So what will you do then? So you can go and do something called ab initio modeling. So in ab initio modeling, what we'll do, we'll do template identification for different region of your query. Okay, let's say 1 to 30 residues can be coming from one template, and 30 to 50 or 60 can be coming from some other template. These templates can be from diverse family members. Okay, it can be just alpha helix followed by beta strand, and then alpha helix, that is alpha by beta fold, or it can be a beta hairpin loop coming up in your structure. So that's the reason why I introduced you to the folds in the motifs in the previous slides. So you can model the different regions of your sequence based on different templates. So you'll model the different regions and assemble them into one complete structure, and at the end you'll do something called minimization. Okay, so minimization or optimization is a necessary step at the end of the modeling, because you need to make sure that there are no steric clashes that are coming up between the side chains of the model residues. Okay, again, ab initio modeling is not supported by all these softwares. Okay, so PIRE2 has an ab initio modeling module inbuilt, and there are two other. One is iTaser, which is an again automated web server, and there's something called Robeta, which is a subsidiary of Rosetta tool. Okay, now, so once you get the structures, you can't just take them and use it for your purposes. You need to evaluate the models. Okay, although every tool or the server provides certain kind of protein modeling quality scores to give the the model protein quality, you have to manually look at your protein structures and do some QC checks, quality checks. So to do this, we'll do some quality assessment based on modeling score is one, and the other one is Raman plot, which is very basically which is regularly used. And there are some advanced methods as well, but right now I'll only discuss about the modeling scores and Raman plot in this session. Okay, and once if the residues that have been or the structure that has been modeled are all in very good quality range, there is nothing to do. You can go ahead and take. But if any residues that are in your structure or the entire model if they are not falling in the respect to quality assessment, you need to do certain kind of refinement. Okay, so this refinement comes up in different stages. One is like you can fine tune the refinement to certain regions, like either refine a loop specifically, or a certain region, or you can refine the specific residues which are undergoing steric clashes, etc. Or even the side chain level refinement can be done. Okay, but the general refinement methodology that one has to use is to do energy minimization. Although the minimization is done at the server level, you also can do further energy minimization after your a model evaluation. So this refinement will be an very high intensive, high resolution and intensive energy minimization refinement that will happen here. So regarding this, I'll also introduce you to two servers which can do this for you. And these kind of energy minimizations can't be done in your local systems. You need high-end systems for them. So these are kind of molecular dynamic simulation based energy minimization refinements. Okay, so regarding the Ramchandran plot and other modeling or quality assessment, I will introduce you to SAFES. And SAFES itself is a server which holds project, and we have something called Rampage to give you Ramchandran plot. There are others also. So today we'll look at the SAFES, which holds the project. And for refinement, if you want to refine specifically the loop regions, we have something called mod loop. And for energy minimizations, we have Chiron, Astra, and mod refiner servers available out there. Okay, so this is the basic protein modeling methodology to go for depending on whether if you have templates or if you don't have templates, you can select the best modeling method, either homology modeling or ab initio modeling. Okay, and you need to evaluate your models for sure after the modeling of your protein structures or the prediction of your protein structures. Okay, so next I'll introduce you to the PIRE2. So we'll be discussing about PIRE2 and Swiss model in the today's session. In PIRE2, first we'll look at the homology modeling methodology. Okay, so PIRE2, as I mentioned, it's an automated server, right? So it has all the steps, pinned together within the backend of its server. So it does everything for you. So you need to just submit the query sequence and wait for the final model to be predicted. And once you get the final model, you'll go ahead and evaluate your model. Although it provides you certain quality assessment, you have to evaluate it out of the box. So now let's see the methodology that has been acquired by the PIRE2 to do homology modeling. So to start with, you have a query sequence of your interested, and then the server will take this sequence and it will align the sequence with the known sequences. Okay, known sequences for which the structures are already known from the experimental studies. Okay, so this can be a database. Okay, so this can be a sequence database or the structural database, which is either hosted by the respective server at the backend, or it can be a public database. For example, it can be PDB. So you can take the sequence and you can align it to all the sequences, okay, of the structures that are there available in the protein database. But the PIRE2 and Swiss model, they have their own databases. These are the public databases downloaded and maintained by the servers at the backend. So the first step is to do a multiple sequence alignment, and then once so for this they use a module called HSB-LITS. So HSB-LITS is a hidden Markov model based search methodology. So which will look for the conserved regions within the sequences. And yes, it will account for the insertions and deletions of the sequences as well. So once you align two sequences, you'll have insertions, deletions, or mutations coming up in the sequences, right? So everything will be accounted for. Now, so once you do the multiple sequence alignment, it will also do something called secondary structure prediction using Cypred. Okay, so Cypred again is another tool which is maintained at the backend. So it's a machine learning based tool, algorithm, which actually helps in predicting the secondary structure for your sequence. Okay, so now you have the secondary structure of your sequence, and also you have the sequence alignment coming up. So what you do at the end, you'll have a hidden Markov model that has been built. Okay, so what is the hidden Markov model? I'm not going into depth of in detail of the hidden Markov models. For now, you can think this as a profile which holds the information with respect to the templates that, the sequence alignment that has occurred, and also the secondary structure predictions. Now, once you have this, it will again check for the coherent template. So we have the profiles that are interchecked between the templates and coherent, and the best templates are picked up, and then it will automatically rank them. Okay. So once you have the templates identified, the next step is to do a crude backbone modeling. As I mentioned, the first step is to model the backbone. So it could be the C alpha backbone conformation. So once you have the template, the scaffold, or the backbone ready, next step is to do a loop modeling. As I mentioned, loop modeling is very difficult because it's very highly dynamic in nature and it has very low sequence similarity. So when you do a sequence alignment also, so there will be a high variation in the loop regions. Okay, so let's conservation. So the next step is to do loop modeling, and once the loop modeling is done, so the tool will go ahead and add the side chains. So once the side chains is added, then depending on certain rotamer libraries, it will just rotate the side chains and make sure that there are no side chain clashes happening up in the structure, and you'll end up with the final model. Okay, and some tools may do energy minimization at the backend, some may not do. Okay, they can have some other optimization methods available. They will change from server to server or tool to tool. Okay, so this is a basic homology modeling methodology that is used by Python. Okay, now next we will also see the ab initio methodology modeling, which is slightly different from this. Okay, that is used by Python. Don't worry, once I walk you through all these methodologies, we will go ahead and submit the sequences to the servers, and we will look at the results, and we'll see, how to pick the best templates, and how when should we pick the multiple templates, and we'll also see how to assess the quality scores, etc. So the next part is the ab initio modeling methodology, and if you see here, the first step is to acquire the normal pipe protocol. So the normal pipe protocol, what it is doing? It is doing sequence alignment, it is identifying the secondary structure, and then it is going for building the profiles. But here it's slightly different because it will do the same, it will apply the same protocol for the different regions of the proteins. Okay, and these regions may be the templates from different proteins. Now, once you have them, once it identifies the different template regions coming up from different multiple proteins, it will put them together, assemble them. Okay, and it will do again, the process will repeat, the backbone and side chain additions will happen. Okay, so the only difference is there you have the modeling happening from one single protein or multiple proteins, but it's only the regions. But mostly in homology modeling, you'll end up getting the protein templates from same family. Okay, but here it can be different, completely different from the respective family. So if you see here, we have completely alpha fold class here, and we have alpha beta class here, and we have some other protein coming up over here. So it can the different regions of your query sequence can be modeled from different sections of different proteins. Okay. Now, so Py2. So once you go to the Py2 web page, this is how the info screen will look like. So you have a few sections here. You need to enter your email address, and if you want, you can enter some job description over here regarding the sequence that you submitted, etc. Or you can paste your amino acid sequence. And there are two modes here. One is normal and the intensive or test mode. Test mode is for developers, so we won't be looking into this. The normal mode is for homology modeling, and the intensive mode is for ab initio modeling. I'll take you there and I'll show you, don't worry. And you need to select for non-for-profit organization if yours is a government or non-profit organization. And if it's a commercial organization, you just need to select the respective option here. Okay, so now let's go back to the Py2 and submit a sequence for homology modeling. Right. So I hope in the previous session, UniProt database is covered. So I'm not going to walk you through the UniProt database. Just please let me know if it is required so that we can cover the UniProt database as well. So Yeah, the sequence that we'll be using today for protein modeling is probable ATP dependent RNA helicase DDX4. Okay, so if I just I'll just show you how to search for the protein of interest sequence. I mean, if you have the sequence with you from your experimental studies, that's well and good. But if you're working on some protein for which you don't have the sequence, this is how you can fetch the sequence. So you need to go to UniProt, and in the search tab, you can just type the respective protein name. So we see here, I have some results coming up over here. Okay, so what I'll do, I'll go ahead and filter them. So from the popular organisms, I select only for humans. Okay, and I ended up with 6 results here. So if you see here, there is something called length information. Okay, 724, 159, 50, 156, 105, 142. Okay, this is the protein sequence length. Okay, now what I'll do, I'll select the first one, which is actually having good coverage. Okay, I'll take this one. These can be the only certain regions of the protein, which I may not be interested in. Most of the time, I'll be interested to look for the entire protein, if not the catalytic domain. Okay, if I just go ahead and select the first result, you see on the left side, we have certain information already given. Like you can directly right away go to the respective information, like the function, name, structure, subcellular population, etc. But the one which we are interested in is structure or family and domains. Okay. Now, if the structures are already available for this sequence, it should see something coming up over here. Okay, see 3D structure databases. If a structure is already available, you should see something over here. But right now, we don't have anything coming up from the experimental point of view. The only one that is available.
Command plus would help.
Yeah, is that visible?
Bit more, if you can afford to.
Yeah.
Yes. Yeah.
So in the structures panel, like at the 3D structure databases, if any structure is already known for this sequence, you will be seeing it over here. That information linked to the database, you will find it over here. But if it is not available, you need to, I mean, I'll consider that, okay, there may not be any experimental data available for that. So what I'll do, I'll go ahead and try to model it. But recently there's a tool called, I mean, it's a predicted prediction tool called AlphaFold. We'll discuss about that at the end. So which, I mean, here if you see, we have a structure that has been already modeled coming up from AlphaFold. For now, we'll skip this. We'll look at this at the end. Okay. And if I go to the family and domains, I have 2 domains in this sequence, I mean, this structure. So one is helicase ATP binding domain, the other one is helicase C-terminal domain. Okay, and these are the positions of that. Okay, in the sequence from 319 to 502, so it is the helicase ATP binding domain, and from 530 to 675, it's the helicase C-terminal domain. So what I'll do, I'll go ahead and select this one. Okay, helicase ATP binding domain. If I click on this, I'll be taken to this page where I can get the FASTA sequence. So this is in the FASTA format. Okay, this is just the plain. So this entire thing is in FASTA format. So I can quickly copy this from here and then go back to the file 2. Okay, so this page. Now, so in any server, so wherever you have certain options given, there'll be a small icon, help icon available. So if you have any doubts, it is with respect to any bioinformatics servers or database or anything. So anywhere wherever there are some options available, there you can see some kind of icons like this, like help or question mark I options. Okay, so if you just hover onto them, it will give you the information. Like for example, if you see here, or the unless, yeah. So if you see here, just beside the amino acid sequence, you have something called I. So if I just play, I mean, hover my cursor on that, I would say either FASTA formatted sequence or raw amino acid sequence is accepted. Okay, only single sequence. But for multiple sequences, you need to use the batch facility. Okay, that we are not doing now. So what we'll do, we'll go ahead and paste our sequence here. This is in FASTA format. So we have a FASTA header over here. Okay, and we have the sequence here. And as I mentioned, we have two modes here. So again, if you hover onto this, it will give you the information. The normal mode is fast and similar to original file, and the intense mode performs complete modeling of your entire protein using multiple templates. Okay, this is basically the ab initio modeling method. Okay, if the templates are not available for your structure. So keep one thing in mind: if you are blindly submitting to ab initio, intensive mode, and if you have templates available for your structure, then it will go with the it's kind of homogeneous modeling there as well. Okay. Now, so what I'll do, I'll select the normal mode. I won't go for intensive mode. So under normal mode, I'll submit. So non-for-profit, and I'll just go ahead and do. Okay. So once I submit, I'll get a job status, and you see whether your job is queued or it is running, etc. But I have the jobs already. I have the job run for this already. It says it takes 1.2 hours plus taken or given 1 hour. So I have the result already for this. So I'll share with you guys the results. So this is how the result page looks. So we have the information with respect to your job, the email ID, your job submission date, and the job ID. And if you want, you can download the sequence that you have already submitted. Okay, and the job type and the job. So you can only avail this information for 30 days. Now coming down, so this is the most crucial information, the summary of your results. Okay. Now this is the model that has been obtained, the predicted structure for your sequence. Now if you see the model left is based on this template. So it has used only one template, that is with respect to this. Now don't worry, look into this, what this template is actually. Now it also gives you some annotation, like it's a translation one and RNA binding protein. Okay, the model we're talking about the template. So the template belongs to translation function property and the RNA binding protein category, and it is the chain C of the respective structure, and the name of the template that has been used, and the PDB title. And these are the three databases where the template structure has been deposited. So RCSB is the PDB. So if you want, you can just go to the respective structure, the PDB. Okay. So this is the reason why I introduced you to the PDB in the previous slide. So if you see here, so this is the template that has been used to model your protein. Now, if you want to know more about your template structure, you can go through this, because if you start from a sequence for which you don't have any clue or any information at all, so depending on this, at least you can fall back onto a structure which has a higher sequence similarity with your sequence, and then consider this for certain studies. Okay. Now, if you're we'll go back again now. So if you see here, we have something called confidence and coverage information. So the confidence for the protein modeling or the predicted structure is 100%. The coverage is 98%. So when we talk about coverage, let's say if you are submitting a sequence length of, for example, if you see here, we have 181 residues. Your sequence length is 181 residues. Okay, the 98% of your sequence has been modeled with 100% confidence. Okay, so this is very important. Whenever you submit your sequence, and at the end when you get a structure predicted, so you need to look at this coverage. Okay, and if required, you can go back and look at the structure where the regions are actually missing, for which it can't predict. Okay. If you want, you can even leave it in the 3D mode, but we'll do it in the PyMod in the local system. So we'll go there. So if you want to download the structure, you can just download the entire results from here, or you can click on image as well. But at the bottom part, we have more important information, like you can get the // pseudo multiple sequence alignment. So if you see here, so this is the multiple sequence alignment that it has performed at the backend. Okay, and the database that has used is UniRef 50, and these are the IDs and the proteins that it has, aligned against. So you have everything, the values, scores, and everything. So if you want to go and get the sequences which are very similar to your query sequence, you can do that. I mean, this also gives you the conserved regions from your sequences as well. Okay. Now we have something called the secondary structure and disorder prediction as well. So if you see here, this is your sequence. Okay. And for yours, as I mentioned, like it uses Cypred to do the secondary structure prediction, right? So this is the secondary structure that has been predicted. Okay, and you have the confidence key. I mean, again, it is fine-tuned by the template identified as well. So you have the confidence key for this, and you also have the disordered region predicted. So whether your structure is in an ordered conformation or disordered conformation, that also we can get this. You can get from this plot. So see here, we have information with respect to the percentage of disorderedness in your structure, alpha helical content, the sheet content in your protein, everything. Okay, and you can also look at the domain analysis, like we have the domains coming from different proteins. These are the different template proteins. Okay, so you can look at the domains that are coming up from the different templates. So you can download and all this report as a PDF again. Now if you scroll down a bit, we have the information with respect to templates that are identified. Okay, so if you see here, there's something called confidence and the percentage identity. So for two structures to be structurally very similar, as I mentioned before, like you can expect 30 to 35 sequence similarity, right? So minimum cutoff is 30 to 35%, and the higher the better, but 100% is like it's the same sequence. So whenever you do blast search, if it has anything close to, I mean, if it has anything higher than 90 to 95, please go and manually inspect your the target sequencer that you might have identified in your alignments. Okay, that could be possibly from the same very closest family, or the same sequence with some mutations or some changes in the sequence. Now if I want, I can actually select certain templates and align them against the model structures. So you just need to select them here, align this model to the master. So I got it already selected few. Okay, and if I just do alignment. Okay. So you can even get the sequence alignment coming up over here and showing. Okay. So you can even do send this model to Pyre mostly to analyze mutations as well. So model quality and Pyre can do more than model. It can do certain investigation to bind, to find binding sites, to look at the mutations or the functional sites that are already known based on the template structures. I mean, it can do a bit of further analysis for you, which we may not look right now. But as I mentioned, like you can select the models or templates, and you can superimpose them onto your main model. See here, most of the templates that are identified are having similar structural conformation. Okay, so this is the backbone. So most of them have similar backbone. So although the sequence identity is low, but the structural conservation is still there, and it is expected. Although you don't find certain sequence similarity, I mean, if the sequence similarity is low, you can find high structural conservation. Okay. Now, so with respect to Pyre, we'll stop at this point and we'll look into the Swiss model, and we'll also have some tasks for you to practice. Don't worry, we'll provide you them at the end. Okay, so we'll skip to the next one, which is called Swiss model. So this is the Swiss model page. So here either you can create, sign up and create a project, and you can have all the files, input and output files are stored alongside the server, and you can work with the project as per your requirement, or you can start modeling without creating a project or logging into the server. So just go ahead and start modeling. So if you see here, this is the submission panel. So it says start a new modeling project. So you can just paste your sequence here and search for the templates, or if you have a template with you, if you have a known template with you, and if you want to submit that as well, you can do that. Okay, so you can just say, you can go to user template and you can upload your template of your interest, or and your sequence of interest, and you can do the modeling. So right now we don't have the template, so we will let the server search for the templates and select some templates for us. So I will go back to the UniProt and fetch the sequence. So you see this job is still running, and it is building hidden mark of the model sequence profiles at the backend. So I'll take the sequence from here. I will submit it over to the Swiss model. Okay. So once you submit, it will take it like this, and I'll just give the project title as test. And if you have any hetero targets as well, you can give them. I mean, the hetero compounds that you have, you can submit them as well. So once I go ahead and search for templates, so it will do the template search, and you can actually see in real time what is happening at the backend. So it will post all the process that are happening.
Please zoom in once. Yeah, that's it.
You see here, it is running HSB list against SAMTL, and it is reusing the cached alignment that is that might have already happened, and so every step, each and every step that it does at the backend, you can actually see it over here. So searching PDB profile database. So this is the database that that it search against. Okay, so it also built a query profile, which it is using based on your sequence. It has built a query profile, and it is using that to search against your databases. Okay, so once you have this done, you will end up with the result something like this. Okay, so once the templates are identified, you will be presented to this result page. Okay, so if you see here, this is called template results. So these are the templates that are identified. Okay, now, so in addition to PI2, this has certain information which can be helpful to us, like for example, quaternary structure. So let's say if the templates have any algorithmic states or quaternary structure available, that information also we can get over from here. So okay, and the sequence similarities. Okay, and so we'll go to the individual template and we'll look at the alignment stone. So if you see here, so this is our target sequence, that is query sequence of our interest, and it has been aligned against this template. Now, if you see here, the secondary structure information of this template is also given over here, right? See helix, we have a strand over here. So that means this sequence can, so from here to here, it can be a strand, and from here to here, this can be a helix when you model when you predict the final structure. Okay. And if you see here, there are certain regions. So this is a gap in our. So if it is a gap in the query sequence, well and good. But if it is a gap in the target, the template sequence, then you need to use multiple templates. So I've been mentioning, right, when to use multiple templates. Okay. So if you see here, this is something called sequence coverage. Okay. Now, in your sequence, for example, if you take this, so certain regions may not be having a good alignment with the respective template. So for example, 80% is aligned properly, but there is still 20% left over. And this 20%, if it is coming from some other template, okay, that is also helpful. But that's just one reason. There's one more, like if you have multiple templates, like the multiple templates which are getting aligned to specific region, So it can be, you can, you have a higher resolution of, information available over there to model the structure. Okay, so it can be used as used in two ways, like to fill in the gaps in your in the missing information, or to get a good resolution of the existing information. Okay, the multiple templates can be used in two ways. Okay, so if you think the coverage and sequence identity or similarity is really good, you can go ahead and use one single sequence for your modeling. And if you think that the coverage is less, and if you and the sequence similarity or the identity is very low, you can go ahead and use multiple templates which can cover the entire range. Okay, so if I just scroll down, so if you see here, these are the gaps in your query sequence, the target sequence. Okay, so if you see here, that's why these are placed below the lowest hits. So these kind of gaps can be filled in by, for example, take this one and this one. So these are the two templates. So whatever missing information it has here, it can be filled in by this one. But the rest is also there. So these regions can provide higher resolution for your modeling part. Okay. So you'll encounter the, these kind of multiple template approaches will be very helpful when you have a lower query coverage or the lower sequence identity similarity. Okay. So now we also have this course, and we'll talk about them in the next modeling step. So we have something called oligo states. So you can see whether it's a monomeric state or it's an algorithmic state. That information also is given. So if you see here, there's one which is a homodimer state. Okay, so this entire information is coming from the respective protein database where they have been deposited. Okay. Now, so if you want to model, what you can do, you can just select the respective templates or just one template. And if you see, I just clicked on this template and it already gave me the structure over here. So I have the three-dimensional structure over here. This is for the template. Okay, this is not the model structure. So this is the template. Okay. Now, so you can expect your query sequence or the target sequence to be modeled into a structure something similar to this. Because this is your template structure. If you go ahead and build models, so it will build a model. So if you see here, this I have already done. So that's what's giving model already exists for the children template. So this is your result page, and this is the place where you have to assess your model structures. So in Pi 2, we are looking at the confidence score, we are looking at the coverage, okay, and we're also looking at the templates that are actually used for the modeling. But here you have a bit more information. So Swiss model actually provides you information with respect to atomic level. So you can go and look at the resolution of your protein with respect to each and every residue. Okay. Now, so let's go by one. So we have a snapshot of your model structure here, and if you click over on this, you can download your data in different formats. Okay, you can download the PDB format of your structure, which can be used further. PDB format is the format in which the structural information is stored. Okay, which is actually the format maintained by the protein data bank. So when you deposit a structure in the protein data bank, so you have to deposit it in certain format. So this format is basically the XYZ coordinates of each and every atom that is present in your structure. Okay, now, so this atomic to load into certain visualizers and then do further analysis. Okay. Regarding this, we'll look at a tool called Pymon. If the time permits, at the end we can have a demo of that as well. Okay. So you can download your structure. You just have to click on it. I've downloaded it in the PDB format. And to submit it for the evaluation servers as well, we need to download the PDB format file. So we have something called structure assessment over here. So if I just click on that, it will give me a Ramchandra plot here itself. Okay, and it has some other tools inbuilt like mol probability results, which will take some time to run. And we have, as I mentioned, residue level of quality. Okay, so we have something called Q mean score. So Q mean score is actually the scoring function that has been incorporated into the Swiss model. So as I mentioned, everything has its. I mean, most of the tools have their own scoring, algorithms. So Q-mean, it will give us the global scoring quality score based on certain formats, like the starting from the template alignment to the structure modeling. So it will consider everything and it will give us some kind of global quality assessment score. Okay, so you have so anything between 0 to 1 is I mean it mostly lies in 0 to 1. Okay, and the higher the better. But if you see here, we have a certain structure here and we have some errors. So if you see, this is the global Q-mean score, and we have atomic level score as well. So if this model is really good enough, you will see it coming up over in the blue region. But it is like slightly in between red and blue. That means we have some errors in your in our putting model. We will see them. We will see where the errors are coming from. Okay, but the Q-mean B is 0.79. 0.79 is good. So overall score, as I mentioned, 0 to 1, you can consider. The higher the better. Okay, so we are getting 0.7. So overall it is good, but you still have some errors. So this is not still good. And if you see here, C beta atoms resolution is poor. Okay, Al atom resolution is good, and the solvation information is also okay. And we have solvation, which so everything is above red except for C beta. So there are some slightly fine tuning that can be done in your protein structures. So when you submit this for energy minimization, most of these issues will be fixed. Okay, so same thing can be seen from here. So local quality estimate and local similarity to target. So with respect to target, how the quality scores are coming up, that also we can see as well. Okay, so this is the Ramachandran plot. If you see here, like small probability results, you are seeing like we have clash score of 4.58. So there is some score given for the clashes that are getting generated after the model is created, and we have something called the Ramachandran favored regions. So if all the residues are present in the favored regions or disallowed regions, that information can be obtained from here. And we have 2.25% of the residues in your sequence are in the outliers, okay, in the outlier regions. So if you want, you can, if you just hover onto these residues, you can actually see where they are coming from. So as you can see, these are in the loop regions. So as I mentioned, right, loop regions are the hard to model regions because these are highly dynamic in nature and more, less conserved in nature. So for the loops, it is, so you see all the residues, these 2.5% residues are coming up in the loop regions. So these can be further fine-tuned, further model using either servers like ModLoop, or you can do it for energy minimization, etc. Okay, and you even have the bad bonds happening, bad angles. Okay, so we have the complete error report that is getting generated here. So you can either be satisfied with this, or you can go for further evaluation using certain tools like saves, etc. Okay, so we'll see what additional information the other tools gives us. Okay, now okay, yeah. So we have the models. So let's go back here. So if you see here, as I mentioned, like the score respective scores, if you just click on this question icons, you can get the scores. I mean, if anybody is interested and want to go through the respective scores in detail, they can just go ahead through this document. So we have the scores given. So G and Q and G is a global. It's between 0 to 1, and the higher the better. Okay, and you have the, for easier observation, you have the scales given as well with the bad and good ranges. Okay, now, so on the right panel, you have the structure that has been modeled, and you can also see the coverage till what extent the structure has been modeled. So if you see here, there is some the C-terminal region, we have some leftover region, so that region has not been modeled. Okay, so you need to manually investigate your structure to see what is happening with the model or predicted structure. So whether we have the complete structure predicted or we have the leftovers left, and you can look at your structure in different representations as well. We can download the structure and use PyMol as well. So PyMol is an standalone software that you can use to explore your structures for further analysis. Okay. Now, so let's go back to the slides. Okay. So are the slides visible now?
Yes, Raj.
Yeah. So this is what we looked at. I'll just briefly recap now what we have looked at. So from PI results, we have looked at the template information section, and we looked at the assessment part. So we'll be looking at confidence. We'll be looking at the coverage of your sequence of interest, like what is the coverage that has been considered for modeling. And we looked at the templates information, and we looked at the predicted secondary structure information, the different secondary structure content like disordered helix and strand content. And we looked at the domain analysis part, and we looked at the Swiss model. So how to submit the sequence for Swiss model, and once the template search is done, so we'll be able to see the complete template identification process over here. And once the template search is done, or how to get the best templates, and whether to go for a single or multiple templates, and whether to select multiple templates or single template, depending on your results, or depending on your interest, we can do that. And you can also view the summary with respect to your templates on the right side panel. And we have the results part, where you have the quality scores and the summary of your model protein structures as well. Then next we will go ahead and look into one of the assessment server that is available online, which is called Saves. So I'll quickly take you there. Hello. Are you guys able to see the browser? Yeah, now I should be able to see. Yes.
You have to zoom in a bit.
Yes. So you see, this is the Saves website version 6.0. The 5.0 is duplicated, so please keep in mind that you need to use the 6.0. So Saves actually holds multiple tools to verify your structures, or to do a fault assessment of your structure. So here you just need to upload the PDB file that you have downloaded. Okay, so I've just uploaded my PDB file. So this is the result predicted PDB structure from the either PIR2 or Swiss model, depending on your interest. You can just upload your predicted structure, and if you just click on Run Programs, it will take you to the page where you have an option to select multiple tools to evaluate your protein structure. So if you see, we have ERAT, Verify 3D, Prove, WatchCheck, ProCheck, and PRIST. So considering the time constraint, I will just walk you through the ProCheck. Okay, and if you see, this checks the stereochemical quality of your protein structure. Okay, so if there are any stereo clashes and this kind of information, it will provide you right away. It will do a residue by residue geometry analysis and overall structure geometry as well. Okay, but if you want statistics or non-bonded interactions between different atom types and etc., you can go ahead and do ERAT. So ERAT also will give you some quality estimate. So I'll quickly walk you through ERAT if you have time. So first let's look at the project now. So I just clicked on start and it is doing some analysis for me at the back end. You can also start the ERAT. Okay, so this results are faster. So before getting into project, I'll also walk you through the ERAT. So if you just go through the results part. So ERAT gives you a quality score, quality factor score based on the assessment it does. So I have shown you like what kind of errors it looks for, and it will also give you a plot something like this. So you have residue level information coming up over here. And if you see, so there's an error value cutoff here, so 95, 99%. So anything below 95 is really good. So you have no bad contacts. You have really good, interactions coming up over in your structure, and everything is good. So if you have anything within 95 to 99%, it is slightly acceptable depending on the regions, for example, loop or anything. But if you want to still fix them, you can again do a refinement of these regions as well. But if anything is above 99%, then those regions need a serious fine tuning. Like you may have to model them again that specific regions, or you need to do extended MD simulations to minimize certain regions or etc. Okay, for now everything looks good. So these are in slightly acceptable ranges. Okay, and if I go to project results, so it gives me something like this. So we have a Ramachandran plot analysis as well. Okay, so I'll take you the plot. That will be easy. Yeah. So we have the project summary coming up over here. So we have something called, so it gives you the number of residues in your model structure and the Ramachandran plot scores. Like, we have 94.8% of the residues in 4 regions of the Ramachandran plot, 3.9% in the allowed regions, and 0 in the general, and 1.3% in the disallowed regions. Okay, and we have more further information with respect to deviations, or deviations from their average or mean, or you have the bad contacts, bond length angles, etc. Okay, so you can also see the planar groups that 89.9% are within the limits, 11.1 are highlighted. So you have the complete information coming up over here. So if I just go ahead and look at, so says Kai2 plot is it has passed this, but sidechain parameters and residue parameters and bond length, we have a little bit of errors coming up over here. Okay, so if I just first go ahead and look at the Ramachandran plot, we have looked at in the Swiss model as well, but people who are not using the Swiss model, if they are just using prior to it, they can come back and submit their structures over here. And if I see here, so we have guys, I'm not going in detail with respect to the Ramachandran plot, but I'll just give you the brief report information over here. So you just need to make sure that most of them are in favored, allowed, or this is the disallowed regions. Okay, so you can clearly see it here. Residues in most favored regions. So this is the most favored regions. So every region corresponds to helices, beta sheet, and the left-handed helices regions. Okay, and so we have 94.8% of the residues falling in the most favored regions. So these two regions are covered, and 3.9% of the residues in your structure are in the allowed regions. So these are allowed. It's okay for them to be there. Okay, but there is one more region. It's generously allowed. Okay, acceptable region. But this is a tricky thing. So there's something called residues in disallowed regions. You need to make sure that at least minimum 2% shouldn't exceed for these disallowed regions. Okay, if you have higher, then you need to go for very fine tuning of your model to make sure that your structure is in really good conformational state. So we have so for our structures from Swiss model, we have 2 residues, that is 1.3% of our sequences coming up in the disallowed regions. So from here you can see what are those 2 residues, that is threonine 38 and glutamic acid 155. So glutamic acid 155. Sorry, glutamic 1. Right. this is glutamic acid, and with threonine we have 38 and 155th position of the residues. Okay, now, so going forward, let's say if you feel that you need a higher refinement required for a structure, as we can see, the loop regions of our structures are not good enough, right? So what we can do, we can go ahead and submit the structure for further energy minimization. Okay, so where can you do that? As I mentioned, we have two servers. One is Asara energy minimization server. Okay, and the other one is mod refiner from Zhang Lab. Okay, so yeah. So first, let's talk about the mod refiner. So mod refiner is it's an atomic level high resolution structure refinement tool. So either you can upload your template structure, only the C alpha atoms of your the reference structure, and the structure that has been obtained from the modeling tool to the server. So what we'll do, we'll try to do some kind of minimization of the backend, and it will give you the best structure with the lowest energy conformation state. So what it's trying to do, it will slightly move the residues in the three-dimensional space, so that they are sitting in the best lowest energy states by avoiding the clashes with the neighboring atoms. Okay. So similar thing happens here in the ASRA. So here you just need to submit your upload your PDB structure that you have obtained, or if you want to minimize any structure that I mean from the PDB also, that you can do directly as well.
Ash, can you zoom in a bit, please?
Yes.
Yeah.
Yeah. So here, unfortunately, you can't submit more than one structure. So you have to submit structures individually. So as you can see here, it is using its own force field to do the energy minimization. Okay. So as I have shown you in the first slide, I mean the second slide. So what it will do, it will slightly apply some force using the force fields, and it will try to see the motion based on the mass and acceleration of the atoms. So they will try to get into the lowest energy state, and which will reduce the steric clashes. So that's what happens at the minimization backend. Okay. So this will take some time. So once you get the structures back from the minimization servers, what you can do, you can go back and submit it to the evaluation tools. It can be the saves. Okay, in our case, so you can submit back to saves and look at the Ramcharan plot or the other steric clashes and the energies. It can be errata also, and see how your structure is actually getting refined in the specific regions. Okay, so that can be done. So we'll leave that part as an assignment for you at the end. So with this, we will stop here with respect to the the modeling servers, and I'll introduce you slowly to the visualizer tool called PyMod. So PyMod is actually visualizer tool which you can use to explore your structures further, in-house, like in your laptop or system, or in your local computer. Okay, so now let me switch back to the slides. Yeah. So are the slides visible?
Yes.
So to recap, we discussed about saves. So we looked at errata and project. In the project, we looked at the summary and how to look at the disallowed region residues. And we also looked at the mod refiner and Astra servers. Then, okay, first, before going to PyMod, I'll also introduce you to a database called AlphaFold. Okay, so AlphaFold is actually the protein structure prediction it's a tool, and AlphaFold also maintains a database for the predicted protein structures. So basically, AlphaFold is the tool from DeepMind, which is supported by the Google, at the backend. Okay, and what it does, it uses a deep neural network based modeling techniques. So what it do, it take the sequence, okay, whatever traditional setup that we are using, finding the templates and and modeling the structures. Okay, so the traditional methodology is actually incorporated into a deep neural network based approach. So what it is doing, it is actually trying to build predictive model based on the known structures. So it has been trained on all the known structures that are available in the databases. So based on the features that have been developed from the known structures, so let's say if you have the dihedral angle pattern, the a stretch of residues are given and their dihedral angles are known, and depending on their charges and the functional features, etc. So given all these features, the dihedral angles, the charge features, the interaction features, etc. So every information is mapped into a feature format. So based on all these features, the neural network, what will do, it will take the sequence as an input, and given these features, it has been tried on, trained and tested on multiple structures. So it knows, like, given this sequence template, what would be the expected structure. Okay, so let's say if the sequence A has a feature set with respect to a charge, a dihedral angle, or and the pattern of the sequence. Let's say 1, 2, 3. This is the pattern, and then 2, 3, 4 again some other pattern. So based on all these criteria, what will try to, it will try to predict the structure, okay, based on the existing knowledge. So these people actually, this tool has actually come up with a really good prediction scores. Okay, so, and it has now been integrated by EMBL EBI, European Microbiology Laboratories, and so this is the data set right now that has been hosted on the AlphaFold. So for the given organisms, so this is the number of predicted structures, okay, for the unknown structures. So it has taken the sequence, all the sequences for which the structures are not known. So the structures have been predicted using the AlphaFold, and all this data is available for download from the AlphaFold. We see for Homo sapiens humans, we have 23,391 structures that has been predicted using AlphaFold. And right now, AlphaFold is considered to be the one of the very accurate structure prediction tool that is out there. So if anybody is interested, they can try out the AlphaFold as well. But these are deep neural network based model, so you need to know how to set up the model and how to use it. Okay, and but this information is only available for the existing sequences. But if you come up with your but if you come up with your new sequence, so if you come up with any new sequence, then you may have to go with the traditional modeling methodology. So the structure is not available in the AlphaFold. Okay, and we can. But then, if we have time, we will explore this as well. The AlphaFold. But right now, I will briefly walk you through the PyMol. Okay, so how to do protein structural analysis using PyMol. So once you get the protein structure, you can go and explore your model protein further using the PyMol tool. Okay, I hope everyone might have installed PyMol already. So these are different, represent molecular visualization representation that you can generate from the PyMol. So the nice, good-looking figures that you see in the papers for the structures are coming from the mostly PyMol tool. Okay, so to walk you through, so this is how once the PyMol is open, we have 4 main panels. One is the GUI and command terminal panel, and the weaving GUI weaver panel, and the something called object control panel and mouse control panel. So if any commands has to be issued with respect to loading the files and building a new confirmation or with respect to trajectory analysis or settings with respect to your visualization, everything can be done over here. And if any commands needs to be typed in manually, so PyMol is basically developed using the Python scripts. Okay, so the backend is basically Python scripting language. Okay, so it's called Python markup visualizer. So we have you can even give the commands over here in this command terminal. So you just give command over here and it will execute it for you at the backend. And whatever you are doing, you can visualize that over here in the viewer. And the controls to control your structure to visualize, change the representations or display or hide, everything can be done from over here. So we have different panels here, A, S, H, L, C. So A is for actions, S is for show, to show different representations or bonds, etc. H is to hide any information that is available on the viewer already. L is to label your information. For example, it can be a residue labeling or it can be a group of residues. Okay, it can be chains, etc. And C is for coloring the different objects in your visualizer. Okay. Now, so to start with, I'll just walk you through the slides first, and then I'll show you the PyMol in real time. Okay. So if you want to fetch a structure from the PDB directly, what you can do, you can just type fetch space and the PDB ID. So as I mentioned, PDB ID is a respect identity number that has been given to a deposited structure in the protein database. Okay, so you can just type that PDB ID over here. So fetch will fetch space PDB ID. What it will do, it will fetch the respective protein structure from the protein data bank. And you can see like this has fetched me this structure. Okay, so and you can see the structure information over here. So we have a structure of human arginine succinate synthetase in complex with aspartate and citrulline. Okay, I mean this. Sorry. So this is this one carbon structure refined at 1.7 angstrom ratio. So I have deleted this one. We have carbon structure refined over here, and if I want to see the sequence of the structure along with the structure here, so what I can go to display and click on the sequence. Okay, so it will show me the sequence here on the top of the GUI viewer. Okay, so you can visualize the sequence, and if you click on the sequence, you can select the respective residues. You will see that, don't worry, in real time I'll show you. Okay, and you can even color the structure depending on different secondary structure content that you have, like it can be helix loop sheet. So you can individually color the different secondary structural regions. And you can even change the representations of your structure. It can be, CPYK, bomb and stick, or ribbon, or cartoon. As we have seen, there are different representations that can be done. And you can specifically, investigate certain regions of your structure. Let's say here I have selected only one specific helix, and I can even look at the side chain polar contacts. Okay, so if I just go to do polar contacts, and within the selection, I can get the polar contacts that are coming up within the selection. So this is my selection, entire selection. So within this, if there are any polar contacts that I can see. Okay, so I'll stop here and then I'll show you the PyMol. So if I just explain everything, it will be too overloaded. So let's get back to the desktop.
Rashikar,
Yes.
Is it possible to give 5 to 10 minutes break to the participants?
Yeah, sure. Sure. Yeah, we can continue after the 5 minutes.
Okay, thank you.
बाइक का तो 2000 रुपये बाइक पूछे नहीं कितने मतलब बाबू पूरा पड़ेगा 2000
So, okay, guys. Now I'll quickly walk you through the PyMod. So, you guys can see the screen, right? So, I have.
Is there a way to zoom it in?
Uh,
Yeah, command prompt section.
much more. PyMod it's difficult.
That's okay. Continue, please.
Yes. I'll go to the screen. So if you see here, this is what I've talked about. So we have a command section over command interface section over here, and we have a visualizer section over here, and we have again the object control section on the right side. Okay, and the mouse mode section, which is mostly used for the simulations purpose. Okay, now first what I'll do, I'll try to fetch a PDB structure with the ID once here, right? So the moment I executed the command, so it has fetched the structure. But see here, so it's an alphabetical protein that I have here. Okay, and all these red star marks that you are seeing are basically the water molecules, and we have some ions as well. So I just go and display the sequence. So you see the sequence is also colored as per the structure. Now these spherical shapes that I have here are these calcium ions that I have here. Okay, calcium ions. Now, so the first thing that I'll do is like I'll beautify the viewer a bit. So I'll go to display and the background I'll change it to white. Okay, and the quality I can increase it to maximum. Now I'll remove the waters because I don't want waters to be in the viewer. So just remove the waters. Okay, now I have the structure. So as I mentioned, A is an action panel. So you can zoom in, center your structure, and look for the origin. Okay, and you can specifically look for certain aspects like a ball and stick model or the ligand sites you can check for. Okay, and you can give certain visualization patterns as well. And in find we have something called find polar contacts. So within the selection or within the specified residue, in between specified residues, you can look for polar contacts as well. Okay, and you can add hydrogens to your structure as well. Okay, and the remove waters, we have already removed the waters right now. And you can, if you want, you can delete the object that you have here. Okay, specific regions you can delete by selecting the regions, or you can delete the entire object as well. Okay, now in the S there is show. So you can show your structure in different formats, like you have wire, line, sticks, okay, cartoon, and spheres, surface, everything. So I'll click surface over here. See, so this is the surface representation of my structure. If you want to hide this, what I can do, I can go to H and then hide the surface. And if I want to change it into some other confirmation like sticks, okay, so I can hide the cotton. Cotton is already there. See, so this is the stick confirmation. So this is easy to look for the interactions. Okay, so stick confirmations are better for to mod. So if I just select on the respective residue, it will get selected. So if I just select a couple of things over here, so this all is selected will come under tab called SCLE. If you want to rename them, you can just rename them saying that okay, my selection, and you can specifically work with this. So if I go to action and find polar contacts within the selection, I can specifically look for polar contacts within the selection. So this is my selection. I will just zoom in. So if you see here, so I asked for the tool to show me the polar contacts. So you see, so I have the polar contacts coming up over here. And I have again selected these two residues specifically. Okay, now if I go back again and go to label, I can specifically label the residues that I have selected as well. So we have alanine 147, okay. And that means 143 we have here. Okay, so similar way you can explore your structure for further. And if you want to look at the active pockets, you can do that as well. If you can make a preset and you can go to the ligand sites. Okay, so it will just take some time. We don't have any ligand sites, but we have the calcium amine sites, so we'll try to get them. What is the previous? Okay, I'll try to show you guys with this around. So this is some other structure that I have loaded. So I can quickly show you some color. I can color them by chain elements or secondary structure. So if I go to by SS, that is secondary structure, I can specifically color them based on the secondary structure. And the moment you color your secondary the structure, your sequence also will get colored as per your structure. And on my so I have selected only one TTP here. I have unselected the one CLF, but I have the both the models are loaded into the tool. So if I go to preset and go for ligand site, so I selected the ligand. So it has changed the structure of the protein to the ribbon mode, and it has specifically given me the active pocket over here. So you see all the residues that are involved in the active site are also represented as the sticks, okay, with a very thin resolution. And you can also see the hydrogen, the contacts that are coming up from the ligand. So this is my ligand over here in the active pocket, and these are the contacts that it is making with the respective residues in the structure. Okay, so you want, I can just go ahead and switch off the contacts as well if required. We can color them to specific requirement if you want to generate the figures. Okay, so if I want to specifically look at the residues that are involved, so what I can do, I can just go ahead, select this residue and see what actually is the residue. So I'm looking at the histidine 86 that is partially involved in an interaction over here. Okay, so this is how you can explore these structures for further analysis. So if you want, I can hide everything and I can specifically go back and look at the cartoon representations. Okay. So I think we are running out of time. We'll stop at this point, and I have given you certain tasks in the slides and in the slides. So we have the commands and certain exercises available. So you can maybe download the presentation and play with your structures, to explore for further information. Okay. So we'll stop here and then go switch back to the slides.
If that's okay, can I take over?
Yes, Srini.
Yeah. Can you stop sharing your screen?
Yeah.
Yeah. Thank you very much, Raj. So I understand that we have done a lot of structure-based analysis so far. So I just wanted to actually take you all through some sort of structure, system-based analysis. So I thought because we have done all of this. So so sorry. Are you able to see my screen?
Yes, sir.
Yeah. So are the slides are visible?
Yes, sir.
Right. So how does it? Okay. Right. So what we are looking at is basically trying to take a completely different view on biological information. What I'll take you through for
You need to switch to presenter.
Oh, so I just swapped the displays. Is this okay now?
Yes.
Perfect. So, so this is what we have seen, and what I want to do is basically take you through the point of view in terms of biological information. So, in the next couple of minutes, I just want to give you an overview of why is bioinformatics so very important, and what is the utility, and what is so very special about this particular discipline of biology. So probably only the bioinformaticians or the computational biologists are the only set of people who can look at both resolution and complexity in information flow. So as you can see here, we have very high resolution. That's what we did so far in terms of looking at structures. You can look at, is my pointer visible?
Yes, sir. Everything
Yeah,
is
yes.
visible.
Perfect. So what we are looking at is basically looking at the resolution of information at an atomic level. We are looking at a lysine and glutamate interacting, and we are looking at the angstrom in terms of distance. So this is nothing but the steps in the pathway that bring about interactions, and so on and so forth. So we can also look at pathways or processes. So it is up to you to look at the resolution of information flow. So this is a cholesterol biosynthesis pathway that we are looking at. And we've all done this in basic biochemistry. You can organize multiple pathways into processes, and gene ontology is a very good way of doing it. And then you can look at the systems levels where you have multiple processes being organized. And then you look at a systems level view in terms of networks. And then we can look at the genome level and at genomes. So what you see here is an increase in resolution where we are looking at an atomic level, and an increase in complexity where the resolution is compromised a bit, but you're looking at the genomes either as part of the population or across species. This is at an astronomical level of looking at biological information flow. So through the workshop, we have looked at different tools that one can use either to look at the atomic level or at the genomes level in terms of QTL mapping across strains and populations, and also doing phylogenetic analysis across species. So in the next few minutes, what I'll do is I'll just take you through how do we look at information at a systems level. So for this, what we use is basically networks. I'll introduce networks to you. So networks are simple graphs. So these are graphs which do not have the X and Y coordinates. So what you look here in terms of the matrix, this is called as adjacency matrix. Let's say that we have two proteins, and this basically tells you the protein-protein interaction profiles. So A interacts with B, B interacts with C, C interacts with D, so on and so forth. Because I have very limited number of items on this list, you can probably look at this and figure out. But if I were to have quite a long list of proteins interacting, then the human eye will not be in a position to actually analyze this piece of information. So how do we then organize this? We organize this in terms of a network. So what do you see here? You have A and B interact with C. C also interacts with D and E. And you can see D interacts with E, C, and F, and E interacts with F and C alongside D. So what we are looking at is basically, this is called as an adjacency matrix. And this adjacency matrix is now converted into a network. So this is exactly what we are looking at. So why is conversion of matrix into a network interesting or important, and what do we understand from this is something we'll see in the next few minutes. So the first measure that we call these as topological measures when you look at the networks. So the first measure that you're looking at is basically called as a degree or connectivity, which basically tells you how many links a particular node has. So this is called as a node, and this is called as an edge or a link. So the line that connects any two nodes is called as a link or an edge. So if we were to look at this, I would say that A has 1 degree, B has 1 degree because it has only one link. And if you are looking at C, C has 4. The degree of C is 4. And again, D, you can see 3, so on and so forth. The idea here is you're just mapping. So if you were to think about your friends network, if you're looking at the Facebook by yourself and you're asking, how many friends am I connected to? You're basically asking what is your connectivity. Now, what does this degree distribution give you? It gives you the probability of finding a node with a specific number of links. And what is really interesting in biological networks is you tend to have very few nodes that are connected to many other nodes, and a lot of nodes are connected to very less nodes. In other way, is what we can say is you have very few hubs. So when we know a person who has a lot of friends, we generally use call him or her as a hub, because if you need some information or a contact, you can definitely fall back on that person. So for this conference, let's say for this workshop, Dr. VLN is the hub, and he is coordinating with the participants, with all the individuals, the instructors, his university officials, so on and so forth. So that is what you see in a biological network. So you have very few hubs and a lot of peripheral nodes which have few connections, right? So that is how the degree works when you're looking in a network, right? So closeness centrality is again another topological measure which shows you the reciprocal average distance to every other node. So we are looking at the shortest link that is required to reach here. So let's say that you have C, you can travel from C to D to F. In case if you do not have this link, then what happens is C to E, E to D, and D to F is the short. You can see the number of nodes one has to go through. This is called as the graph distance. So number of nodes that you actually jump for the information processing. So what do we know about closeness? Node with highest closeness or high closeness is close to all other nodes in terms of graph distance. So by graph distance, I'm looking at 1 link, 2 link, 3 link. So if I have to travel from A to E, I can look at the shortest graph distance that I have. So A to C, C to E, and E to F, for instance. So this is how it happens. And the moment you have a mutation, let's say you have a mutation here, so this link is lost, then you have to go through the entire network. So this is where we see that mutations tend to manifest phenotypes, but they might not be detrimental because you're losing the shortest path to travel. Now, between the centrality, for you to kind of give you an idea, you know, every city has one particular junction where you always see that there is a huge traffic jam. Let's say that you have to reach to a bus stand or a train station. You see that there will be one junction which basically chokes up. For instance, if people in Tirupati might quickly connect to Leela Mahal Circle. So what happens here is this has the highest betweenness because a lot of links tend to converge there, and then this becomes the choke point. So if I were to block this particular, in this case C, if I were to block this or remove this, what happens? A and B fall off, and D, E, and F would fall off. There is no connectivity between AB and this module called as DEF. So that's where between a centrality becomes really important. You're just looking at network and trying to figure out what is the importance of a node and what does that actually mean, right? So nodes that occur on many shortest pathways between other nodes have high betweenness, and these are the ones, if you have to really talk in terms of biology, the nodes that are targeted by pathogens, because they don't need to target the entire network. They can target these choke points and say, I'm going to hijack this protein through a molecular interaction so the network falls apart, and I can hijack the entire system for my own replication. So viruses, for instance, do what is called as molecular mimicry, which end up hijacking the hubs. So if you're looking at host pathogen interactions, please figure out what is the importance of the protein for the physiology of the host that the pathogen is hijacking by interacting with it, right? So this is something that you can actually bear in mind. So finally, what I'll do is I'll take you through clustering coefficient. So clustering coefficient basically gives you the cohesiveness of the neighborhood of a node. So which basically tells you how close the neighborhood is. For instance, if you're looking at C, D, E, and F here, you can see that it is very tightly linked. There is only one link that is missing. If I were to draw a line from C to F, you will see that C, D, E, and F are very tightly connected, and you have each node interacting with every other node. So a good example for such, we call these as cliques. A good example for such clique is a protein complex. What do you see in a protein complex is basically all the members cohesively packed, and this brings about high clustering coefficient, right? So I'll take you through context dependence and functionality. In this case, we are just going to look at the degree. So in both the cases, the blue nodes that you see in the center are the ones which have good number of interactions. And let's just count 1, 2, 3, 4, 5, 6, 7, 8 interactions this blue node has. So we call this as a hub. And then you see this blue node in this in the right side figure. It also has 8 interactions. The only difference in both of these cases is this is an intra-community hub, which basically means that it is something like a protein complex, and you see that it is connected with everything. And in this case, it is called as inter-community hub, where it communicates with different communities. So this is a community. This is a community. So you have blue as a community and the yellow as a community. And this is the hub that we are looking at. In other words, this has very high betweenness. Though the degree for both of these nodes is the same, you can clearly see that this has high betweenness. So for instance, this can be a process. This can be a process or a complex. So what this is doing is bringing about the crosstalk between different processes. And you can think of a kinase. Kinase is a very good example because it phosphorylates multiple processes. So it kind of interacts with a lot of substrates belonging to different processes and pathways. And the moment you kind of block this kinase or hijack this kinase, you know that this crosstalk is completely abrogated. So I keep repeating this. This is something that we have to bear in mind when we are looking at host pathogen interactions. Just look at what is the physiological function of the protein, the host protein, which is interacting with the pathogen, because the pathogen always tries to hijack the host proteins. So we'll move on to the next one, where if anyone who is working on mutations, we always talk about genotype to phenotype links. So we can do this by looking at networks. So here is an example for you. You have the protein A, which has a lot of interactions, you can see. And what happens essentially with protein A is if you have a mutation, and this mutation completely abrogates this protein, so it is not stable any longer. The protein collapses and gets degraded. So all these interactions are lost. So this is what we call as nodal perturbation. And in nodal perturbation, what happens is there's no protein or no active protein which can bring about the interactions. So therefore, what you end up doing is you end up missing all the interactions. Now, this kind of nodal perturbation often leads to a very strong phenotype. So when you're doing experiments, when you're looking at certain aspects, you start thinking about, oh, I get a phenotype, and then I start looking at what is the molecular basis of the phenotype. So for you to understand this, you often end up with a very strong nodal perturbation. You can also have a scenario where you can have an energetic perturbation. So you have a mutation. The mutation is going to kind of abrogate only one interaction, and all the other interactions stay alive. And more often than not, it manifests a relatively lesser phenotype. However, if B is a hub, right, and this A is also a hub, and both these hubs are getting connected, you tend to see that this one interaction can also lead to a very strong manifestation. So this is something that we have to bear in mind when we are looking at what kind of perturbations do these cause at a network level. Right. So I just wanted to give you a brief account of what we talk in terms of networks, because more often than not, when you look at systems biology papers, they talk in terms of network, and they all look good. But how do we interpret, and what does this actually let you know, is something that we have to work on. Now, here is a simple regulator target network. So you see that you have a regulator in yeast which has been deleted, and then the gene expression changes. I'm just giving you only toy examples so that we can appreciate this much better. So what we are seeing here is basically you have a regulator mutant, and then gene expression changes have been studied. And this is a very good paper in Cell. So they have knocked out about 500 or so regulators, and they started looking at gene expression changes. The point that I want to drive here is one on this. Let's say we have mapped the gene regulatory network. So this is how it looks like. So there's something that I want you to kind of have a look at. So when we started with our analysis, we never used arrows. We were talking about blank networks, which links which did not have any arrows, because in this adjacency matrix that we are looking at, we said it's a protein-protein interaction. We don't know whether A binds to C or C binds to A, so there is no arrow. Such graphs are called as undirected graphs. The moment we start talking about regulatory networks, we are talking about A regulating B, for instance. Therefore, there is, in this case, you can see B regulating I. So B becomes the regulator and I becomes the target, and therefore you have an arrow here. So these are called as directed graphs. So if you are talking about any regulation, it could be for phosphorylation, it could be post-transcriptional regulation, let's say an RNA binding protein binding to an mRNA molecule. So you start looking at these as directed graphs, and what you have is basically a regulator and a target. Now, all this is good, but what is really disturbing in this is we are not able to make any sense out of this, right? So can we reorganize this network in such a way that we'll start making sense out of it? So we are not doing any analysis here. We are just looking at the possibility of reconstructing the network in terms of visualization, going for something like this. So it's the same network that we looked at, but the moment we started organizing this, you see that you have a super regulator here, or the master regulator. So A regulates B, C, D, E, and F, E, and then you have a lot of these interactions. So you have tier 1 regulator, tier 2, tier 3, and then you have all the targets. So what we have done is we have organized this in a hierarchical manner. So just to kind of give you a feel for what we have looked at. So this is the network that we generate. And what we have done is aligned it in such a way that it is now hierarchical in nature. And what you look at is basically a hierarchical network, which gives you what is a master regulator, tier 2, tier 3, so on and so forth. So if I were to hit A, this basically means that the entire network collapses. So you can see that, this can lead to a developmental anomaly. So we talk about fetal abortion, so on and so forth. So these could be master regulators which get affected. And then you see that, there could be lethality at the zygotic level, embryonic level, prenatal, postnatal, so on and so forth, depending on the importance of the molecule that we are looking at. Right. So here is an interesting case where we are looking at protein complexes. So we talked about crosstalks. So SNF5 is a chromatin remodeler protein, and we were interested in looking at polyglutamine stretches and what kind of interactions do they do. And the reason that I put up this network is to show as to how much amount of information that you can load onto a network, right? So you have the edges are colored. So you can see that this is a different media, and YPD stands for glucose, and this is a glycerol media, and the orange ones are found in both the carbon sources. And you can see that the edges clearly show which media that we are looking at. And then these are complexes within themselves. So we are looking at the risk complex. You can see 3 members lose interactions in the glycerol YPG medium. You have the 3 green ones, which means that in glucose, you see that RNA polymerase 3 interactors lose interactions, so on and so forth. Now, what is also interesting in this case is you're giving the name of the interactor. You can clearly see what is the gene symbol that is there. This basically gives you the interactor name. And you also have the name of the complex. So if you look at the name of the complex, what you see is basically the function that this complex brings about. So if I'm looking at dark blue, these complexes basically participate in DNA binding and chromatin assembly. If I'm looking at the orange ones, these are ribosome biogenesis and protein synthesis. So this is just to give you an idea as to how much information you can load onto a network and how effectively networks give you a lot of information. And you can keep discussing it as you go along, right? Now, the point that we are discussing here is we are not looking at the entire protein. We are looking at only one region within a particular protein and what are the interactions that it brings about across all the members of different complexes. So I'll also slightly take you through. This is one of the landmark papers that came in 2010. So this basically shows you the genetic landscape of the cell where 2 genes were deleted in yeast together and people started mapping. So it's not protein-protein interaction, it is genetic interaction. And what they figured out is basically when you look at clusters, this is what we were talking in terms of clustering coefficient. These clusters, when they overlaid with biological process information, they could clearly see that they could identify all the processes and their underlying interactors come up. So again, this is basically gene deletion work that has been done. and you can please see as to how networks can help you visualize the underlying phenomenon. And again, for any two processes, you'll again see a lot of molecules that bring about crosstalk. Right. So until this point, what we looked at is basically, the same type of nodes. So whether we are looking at on the transcription factor targets, we were looking at the protein-protein interactions, we are looking just at the genes or the proteins. Here, what we are looking at is two different aspects being connected. So such networks are called as bipartite networks. So until now, we were looking at the unipartite network. It was largely about genes. And here you have two distinct characters. So I don't want to get into the biology of what has been done, but the point here is you have proteins here, and then you have the biological processes. So for each protein, we are mapping it to biological processes, and you can clearly see, again, it's the same thing. Green stands for the glucose, orange for the glycerol. And if you have both, it has been shown with a half circle color coded with green and the other half with red. So what you see here is basically the biological process, and the size of the node actually gives you how many number of such proteins are linked to that particular process. So if I were to look at this image, I can quickly tell you that metabolism is very heavily affected, and transcription and chromatin remodeling. This doesn't come as a surprise because SNF5, we said that it is a chromatin remodeler protein. So you can see that it affects a lot of chromatin remodeling function, so on and so forth. So such bipartite networks, I mean, if you're looking at your data that you have generated, you want to actually map and then figure out which processes are getting affected, you can start thinking in terms of organizing them as networks, because it is one, easy for visualization, and two, if you want to do a downstream analysis, that oh, I can look at a chromatin remodeling as one of the functions that I want to test, a transcription or metabolism, depending on whatever assays that you have to follow up, right? It is also important that you clearly describe each one of them in your text when you're writing a paper, rather than saying that, we see these popping up, because it's not a surprise. I can look at your figure and say that these are popping up. So what is the effective take home is something that one has to discuss, right? I'll just take you to another landmark paper that came in 2007. This is from Albert Laszlo Barbasi's group from Harvard. So first time what they did is they started organizing the phenome and the genome with respect to the diseases together. So this was way back in 2007. And from there on, the understanding of a different biological phenomenon in terms of bipartite networks has taken off. So what you see in this case is basically diseases which have been organized, and these are disease genes, right? So the size of the diseases actually tells you the number of genes that underlie this particular disease. For instance, breast cancer has way too many diseases, way too many genes, and something like spastic ataxia, paraplegia has very low number of genes. And the colors, again, we will not get into the detail of it, but technically speaking, they also give you which organ systems and what kind of disease this actually affects. Now, this is a bipartite network because we are looking at the genes and we are looking at the phenotypes. Now, from this bipartite network, one can actually generate unipartite networks. Again, you are connecting any two diseases if they share a same gene. That is what the human disease network is all about. So you can see that spinal muscular atrophy is connected to a few diseases, which means that it shares the underlying gene. Now, why is this important? This is important because you can see comorbid diseases coming up, which means that if I have obesity and type 2 diabetes, you see that they'll be linked because they have the genes underlying, and a lot of new connections also popped up in this analysis, which people were not aware of prior to this, clearly showing that two diseases can coexist because they have a common gene underlying it. Now, in the right side, you have the same representation for disease gene networks, where any two genes are connected if they participate in the same disease. Now, this is again the same thing that I've shown you there with much better resolution. So I would definitely recommend you guys to go and have a look at this paper and understand. You can see how the disorders have been classified, and the node sizes give you how many number of genes have been identified there. So because I'm running out of time, I'm quickly shifting to using Cytoscape. So I have another 10 minutes to go. So what I'll do is basically stop my presentation here. And I'll just share my Cytoscape. So I hope you've all downloaded Cytoscape. Cytoscape is a wonderful network visualization and a network analysis tool. So before I go there, I just want to give you a quick overview. So I hope you are able to see my screen. Right. So you would have downloaded Cytoscape from here. So I had sent you the download link. And what I want you to also appreciate is basically, is my screen visible?
Yes, really.
Yeah. So
Yes.
yeah. So you have a lot of apps in Cytoscape. So I'll give you a brief account of Cytoscape very quickly. But you can see that you have a lot of apps, which gives you, I mean, these were called as plugins previously. Now these are called as apps. You can click on these apps and kind of figure out what is it that you would want to do. If you want to identify clusters, you can go and use clusters. If you want to actually identify biological processes for clusters, map biological process for clusters, you can do it. You see, you have Clugo and other ones that you can bingo that you can think of, which identifies overrepresented Go terms in the network and display them for you. And you can also organize or visualize things according to your, what you think is the best representation. That's what networks do for you. So just dumping in a lot of information in networks is not helpful, because nobody is interested in seeing a highly dense network. So if you want to discuss the biology underlying something, please ensure that you give the best representation. You can keep playing around with your networks, give your best representation, and start discussing biology underlying what you're doing. Rather than saying that I've done this A analysis, B analysis, I just want to throw some networks in and say that we have also done it, because it's not very exciting. And then you can also look at encode. It gives you clusters based on topology to identify densely populated clusters. Now, the point here is I haven't downloaded any of these apps. This is for you to just go and give a shot. If you have network, you can keep generating networks for everything that you're doing, and you can start looking at it. So what I'll do is basically I'll stop this, and okay, I could have continued. Right. So I'm just wondering if you're able to see my Cytoscape.
Yes, sir, it is visible.
Perfect. So Cytoscape is a tool. I mean, I've just switched it on. You guys can try as well. So this is how your home looks like. I've worked on some of this. So this shows you recent sessions, and these are the sample sessions that you have. You can just load whichever session that you want, and in the task that I've given, you'll have the East Transcription Regulatory Network. That's what is called as the East TRN. So East TRN is basically a directed graph. So when we are analyzing it, you should have the directed graph. So I'll just directly go to this network. So once you see this this is a HIV human protein-protein interaction network. This has 471 nodes and 917 edges. And you can also save this as a .cys file and keep sharing across different people who are working on the project. So these are basically custom made for collaborative work, and you can just go ahead and do. Let me actually do this. I'll just generate this this network. So let's just give a couple of minutes. So the East gene interaction network that I showed you, you have a demo for this. You can just play around with the demo. And you also have command lines that you can use if you're really ventured into this. So what you have here is basically a network that has been placed, and you can annotate your networks. Okay, so you can use different styles. You can color the nodes. You can color the edges. You can label them. You can go about working on their font sizes, so on and so forth, for bringing about real good representation of your networks. And what you can also do is basically change the layout. So I'm just going to give you a quick, so this is the layout that you have. Suddenly you have changed the layout based on a particular parameter. So you have to play around with the layouts. You have a lot of options, but for the want of time, I'm not getting into the details of it. So some of them would take quite a long time. Hierarchical layouts. So this is going to take a bit of a time. So I'm just canceling it for the time being. So you can go, depending on what you're looking at, you can just look at the layout that really helps your case, right? So let me just check if, so this is the one that we started out with. Now you can clearly see where are the hubs. The hubs have been identified here, and you have the peripheral nodes connected over here. So the idea is I'm happy to have this kind of network, but I want to identify what is the degree for each one of them, what is the clustering coefficient, closeness or betweenness, so on and so forth. So what do? So I just go to oopsie, right. So you can just analyze your network in tools. You just say analyze network. So the first thing that it is going to ask is this a directed graph or is this an undirected graph? If it's a directed graph, you will have in degree and out degree. So out degree, so degree is the link between any two nodes. And if you have out degree, it means that it is a regulator from which you have the arrows radiating out. And if you have in degree, it is going to be the target towards which the arrows converge. Now, I'll just consider this as a directed graph. If it is an undirected graph, you will not have in degree and out degree. You'll just have the degree as the component. Right. So what we are looking at is basically this network, and we'll click on the node table. So when we're looking at the node table, here you have all the information that you have, right? So you have the name, you have whatever information has been provided. And what is really interesting for us is we are looking at the average shortest path length, the clustering coefficient, the closeness centrality, right? So you have eccentricity, stress, edge count, and then you have the in degree and out degree. So if it were to be considered as an undirected graph, you'll not have in degree and out degree, you'll just have degree as a component. And then you have between a centrality, neighborhood connectivity, so on and so forth. So you can simply use this to identify what are the important genes that you're looking at in your network, and then come back and say, okay, how is this relevant to what I'm doing? Right? For instance, if you've done a proteomic study and you have a very good interaction network available for you, so you can immediately work on this and figure out what does this exactly mean for whatever you're investigating. So organizing your data in the form of a network and figuring out what are the nodes that are important. And if you're doing mutation-based studies, you can ask which edges are lost. Is the node completely lost? If it is lost, what are the interactions that are completely lost? So on and so forth. Now, with this, what I'll do is I'll let you play around a bit because we are nearing the close of the session. But before I do this, I would tell you to go just figure out what layouts work for the kind of data that you're looking at. So for the East transcription regulatory network that I've given a task, just give it a shot. Figure out what is it that you would want to do. Have a look at what is the best way to visualize. And as I showed, please do not have the habit of just showing networks in your papers, because it doesn't make any sense unless you overlay biology onto it. So this is something, please, this is for all the researchers who want to publish a paper immediately. Please ensure that there is biology to your networks, otherwise it's no longer interesting. Right. As a final point, just because we started with structures, I thought, is my Google Chrome visible?
Yes, sir.
Yes, perfect. So we started with structures. We started. Sorry.
Yeah, your browser is.
Yeah. Thank you. So we started with structures. So the idea here is, can I visualize a structure in the form of a network? So that's a possibility. Here is the tool. This is called as the protein contacts atlas. So what you do is you can just give, there are a few examples if you want to play around. So all that you have to do is give a structure. This is the PDB ID. So I'm not going to go back and explain anything on what a PDB ID is. So you get this information. And once you feed this in, I've just done one particular beta-2 adrenergic receptor. So what you see is basically the chains that you have. So and then you have the secondary structure elements with the sequence provided. And I don't know whether you've seen, when I'm hovering over the mouse, you can see that a lot of things change in the card plot. So what does this mean? We are talking about the interactions between any two structural elements here, given by the colors. And when we keep moving it, you can see as to how exactly the interactions vary. So you can also have a connection with this particular plot. I'm not going to get into the details of it. You can export this for Cytoscape to analyze what's happening. So here is the residue. This is an asteroid plot, which gives you what is the residue with the maximum number of interactions. You can hover your mouse and get connections done. So it's going to take a bit of a time. So I just moved on to one particular residue, and you can see that in A chain what's happening. And you also have what is called as a per residue statistics. Again, we are talking about structures. We are converting them into networks here. So you have the solvated area, and you're talking about what's happening with solvation. You have degree, betweenness, closeness. So it is not as though you can apply networks only to something as big as a protein or a protein-protein interaction. You can start looking at your structures, convert your structures into networks, and start looking at these residues to figure out which residue is important, or what kind of interactions does the residue bring about, and why is it that if it mutates, the protein collapses or a particular interaction is lost, purely from a network perspective. Now, to kind of finalize or finish what we are discussing, all that we are looking at is basically static networks, right? So you have protein-protein interaction mapped at one particular point in time, or people have assembled it over collecting information from literature. So these are static networks. So what we have done more recently is to look at dynamic networks. So if I say that we have looked at dynamic networks, you might be wondering what am I talking about. If I were to use the word contact tracing, that is called as dynamic networks. So what did we do? If there was a spread of COVID in a certain area, we started tracing who contacted whom and from whom did one acquire the infection, and started mapping them and isolating them. So that is what is called as a dynamic network. So you can use this in telephonic communications. You can use, this was first used for tracing sexually transmitted diseases to figure out as to how STDs get transmitted. And during COVID, we had dynamic network analysis being applied very effectively. So this is just to give you an overview. Right, I'll stop here. We will take questions because it's been Raj and I talking all through. So if the audience have any questions, we'll be happy to answer. So this is for you guys to go back and explore a bit. If you have any questions, please feel free to ask now or write to us. We'll kind of look back on your questions and then work with you. Right. So any questions, please? So I see that there is one for Raj. What must be the minimum similarity percentage between template and query for homology modeling? Raj, would you want to take this?
Yes, Srinivas. So for homology modeling, as I mentioned, it should be minimum 30 to 35%. The higher, the better. So you would like to consider the sequence similarity between the template and query with at least minimum 30 to 35% sequence similarity. So the higher, the better.
Right. So any other questions? Well, I know that we'll have a lot of you can play back on this video.
Sir, one more question there in the chat box.
So how can we draw for differentially expressed proteins for PPI? Right. So I don't know where does this question stem from. So when we are looking at differentially expressed proteins, you have two different systems in place. What you can do is, if it's an organism for which, like, you're working on something like an yeast, humans, or maybe Arabidopsis, the model systems where you already have protein-protein interaction networks, you can overlay all your proteomics data onto it to figure out what proteins are expressed, and then you ask which of the nodes are actually differentially expressed nodes. So that will give you an idea as to what exactly is happening with respect to your network. So when you say differentially expressed, so in one condition, you might have certain proteins being overexpressed. In other conditions, you can also have certain proteins being, let's say, downregulated. So that would mean that, I'm just using some terms from biochemistry here, that the stoichiometry of interactions. Sorry. So the stoichiometry of interactions completely change. And then you say which cluster or which pathway or process is getting upregulated and which is getting downregulated. You can use something like a clue go in the Cytoscape to actually map this. Oh, what is the platform for dynamic networking? Right. So in biology, thus far, we have not been doing a lot of dynamic networking. Cytoscape largely captures static networks. So we normally end up writing our own codes if we have to look into dynamic networks. I mean, I haven't used a platform per se so far, but you can definitely, I mean, I haven't looked into platforms because I write my own codes for them. It's easy, like, you know, you have packages inbuilt in Python as well as, so iGraph is a good package for you to use if you're using R. But the data has to be a time series data. So if you do not have time series data, then dynamic networking doesn't make any sense. Right. I think we are just well over near the time, because I was told by VLN that we have to wind up by 5. So if I've rushed, I mean, you'll have access to videos. Please do play back and see. If you have any questions, please write to us. We'll be happy to respond to you.
Yes, sir. Feedback form is there, sir. If any specific things are there, it is going to come, sir. We generated now, sir.
Yeah, yes. I have circulated it around. So we should get information on that. Right, I'll stop here because we have.
Thank you very much, sir.
Thank you.
Over to VLN, sir.
Thank you, Dr. Srinivas and Dr. Rashekar, for a nice presentation as well as the demonstration about different software, Cytoscape and other protein database and so. So actually, my associate deans are about to come, but he has some other meeting with Dean of Agriculture. So I just want to give summarize the workshop and my concluding remarks, and followed by the vote of thanks by Dr. Amaravati. So I hope the workshop was very useful, and expecting to enhance your expertise in the field of bioinformatics. To see the summary of the workshop, actually the total number of registered participants are 360, but registered and paid participants are 268. But if you see the number of participants are attending, it's around 100 only. But anyway, it may be for various reasons. However, as organizers and as a resource persons of the workshop, we hope we gave our best to give demonstrations and explaining the concepts and clarifying the several doubts raised by the participants. So as we promised, we already shared our recorded videos to all the participants, particularly through YouTube, and except today's. So today's also we will upload by tomorrow. And in addition to that, we will provide the e-certificate of achievement and the workshop manual and PPTs of each session, maybe in a week's time maximum. So I request you all to fill the feedback form, which I already posted in the chat box.
Good evening, sir.
Yes.
Sir, my name is Rajendra, sir, working as a SRF in IMR.
Yeah, sir.
My request is to please conduct any workshop regarding this statistical analysis in R, or and second one is in the NGS data analysis in Linux system.
So
if possible, sir.
Yeah,
future.
that we will think of.
Yeah.
Okay, this is just the beginning from our department, because ours is a newly.
I think Supriya ma'am is expert in NGS. I hope.
Okay, that we will see.
Okay.
Somebody is telling videos are not received, so I request you to see in your spam.
Yes, sir.
Because I asked many of my students, they said even mobile phone also, those videos are opening. So you just see if any problem is there from your end, and otherwise I'll try to send in a different format those who have not received. So I request you
Okay,
all to fill the feedback form, which I already posted in the chat box, so that it gives you whatever suggestions you are going to give, that really helps us to improve our upcoming workshops in the near future. And if you have still any queries to the technical sessions, still that questionnaire is there. We keep that questionnaire 1 week valid. After that, we will be deleting. If you send the question, then I will request our resource persons to address your questions. Okay, so that is from my end. So now I request Dr. Amravati to give a formal vote of thanks.
Thank you, sir. Good evening, everyone. Myself, Dr. Amravati, course coordinator, working as assistant professor in the Department of Molecular Biology and Biotechnology, Engineering Agriculture University, College, Tirupati. I really feel honored and privileged to get the opportunity to propose a vote of thanks on this special occasion of online workshop, that is molecular data analysis through bioinformatic tools, organized from 1st to 10th of November 2021, organized in Diamond Jubilee year of the SV Agriculture College, established way back in 1961. I immensely thank the resource persons, Dr. A. Maheshwari Madam, Dr. Sujan Mamiti, Dr. Vaibhav Vindal, Dr. Supriya, Dr. Srinivas Chavali, Dr. Rajshekhar, Dr. VLN Reddy sir, and also myself, for enlightening us with their knowledge and rich experience in their areas of expertise. Each expert unraveled deep insights into the topics and also provided hands-on training with each of the bioinformatic tools and softwares, like accessing the genome sequence data from the GenBank, NCBI, UniProt, blast analysis, gene prediction by GeneScan, SSRIT, MESA, primer design with Primer 3, Primer 3 Plus, microRNA analysis by MIRBase, MFold, protein structure prediction and network analysis by employing PyRe2, Swiss model, and Cytoscape softwares. And also, two experts, they dealt with population analysis like linkage mapping, QTL analysis, and genome-wide association mapping by Carthagene, Eugene, and Tassel, and also phylogenetic and population structure analysis by Mega, Darwin, and structure softwares. I'm pretty sure the precious knowledge of the eminent speakers definitely helped the participants in understanding the significance of bioinformatic tools in analyzing the molecular data and their subsequent application in medicine, agriculture, animal husbandry, and biotech industry. Once again, on behalf of the organizing committee, I convey deep regard and hearty thanks to all our speakers for taking time out from their busy schedule and enlightening us with their knowledge. I'd like to thank all the honorable delegates who blessed us with their presence. I hope you all have enjoyed the workshop. I, on behalf of the organizing committee, convey profound regards and gratitude to our Honorable Vice Chancellor, Sir, Dr. A. Vishnuvardhan Reddy Garu, Dean of Agriculture, Dr. A. Pratap Kumar Reddy Sir, Dean of PG Studies, Dr. G. Ramarao Garu, Registrar, Dr. T. Giridhar Krishna Garu, and other university officers for giving permission to organize this workshop. My sincere thanks to our beloved Associate Dean, Sir, Dr. B. Ravindranath Reddy Garu, for various helps we received towards this workshop. We will be ever remain grateful to you, and we thank you for being with us today, sir. I am very much thankful to Dr. D. Loknath Reddy, sir, University Head, Department of Biotechnology, all the faculty colleagues and non-teaching staff members who always stand by us for making this event a successful one. I'd like to thank our head of the department, Dr. VLN Reddy, sir, for his constant help and guidance that radiated a source of energy within us. An event of this dimension cannot happen overnight. The wheels start rolling months in advance. We have been fortunate enough to have a team of very motivated and dedicated students at the Department of Molecular Biology and Biotechnology. I'm very much thankful to everyone for their involvement beyond their comfort zones. I would like to extend my thanks to all the students and staff of our university and other universities, institutes across the country and abroad for their participation and to take on the accomplishment of this workshop. Thank you is such a prayer that cannot be seen or touched. It must be felt by heart. So thank you once again, all of you. Okay, signing off from my side.
Thank you very much. If any issues are there, you please mail me. We'll try to address this. Okay, I'm winding up the session. Thank you all.
Okay, thank you, sir.
Thank you.
Thank you.
Thank you, sir.